Hello and welcome everyone. In the last class, we discussed about the different assays that researchers perform to get information about the different aspects of the chromatin. So like they can perform nucleosome positioning assays to get known about the positioning of nucleosomes for getting information about the accessible or the open regions of chromatin. They perform chromatin accessibility assays like DNA's one hypersensitivity assay. We discussed about this in details in the last class. Again, there's ATAC sequencing. We'll discuss about this in upcoming video lectures. So there are other types of assays like histone modification assays and chromatin conformation assays for getting information about the 3D architecture of the chromatin. Okay, so today's topic of focus will be nucleosome positioning assays, in which we will be discussing about the MNase-qPCR or MNase sequencing, as well as altered restriction enzyme access on a linear array. So we have already discussed about the functioning of MNase, how MNase works, but let's just brush on this topic. So MNase means micrococal nuclease. This is an endonuclease which can cut DNA non-sequence specifically. Means it doesn't require sequence specificity. It doesn't have to recognize any sequence on the DNA. It can bind to the DNA of any sequence and it can cleave it. But the here comes the fact that the MNase, for MNase to cut the DNA, it must surround the DNA. So here in the schematic you can see the MNase is surrounding the DNA like a grip, like a clamp. So it must surround the DNA completely in order to cleave it. So this is only possible in the case of the DNA which is not occluded by nucleosomes, which is free. So that's why MNase can only cleave the linker DNA, and that is why when we digest the chromatin with MNase, we will see fragments, repetitive fragments of repetitive size of certain size. Okay, like for example, will we will be only getting either getting mononucleosomes, we'll be getting dinucleosomes, trinucleosomes, so on and so forth, right? So whatever we are going to get will be repetition of 147 base pairs. Okay, we have already discussed about this. So how do scientists use this MNase for for getting information about like whether the chromatin is occluded by nucleosomes or not? So let's look at it. Okay, let's say this is the slab of gel. Okay, this is slab of gel, and there are two wells, well A and well B. Okay, in well A, you have put the DNA ladder, which gives us size markers. Okay. Okay, so this is the DNA ladder. In the well B, you will put the chromatin, which is digested by MNase. Okay. So once let's say that the resolution of this gel is 200 base pair. It can't resolve more than that. So this is the maximum resolution of this gel. Okay, so let's say it's so the size, the smallest size it can resolve to is 200 base pairs, and this is, let's say this is 300 base pair size, this is 400 base pairs, this is 500, 600, so on and so forth. Okay, let's just assume that this is a hypothetical situation. So once you digest the chromatin by MNase, what you will be getting? You will be getting the DNA fragments which will be size 200, 400, 600, 800, 1 kb, and so on. Okay, so this will be the way that will be getting the fragments of the DNA. So what you can do, you can for knowing, so let's say that you want an information that you are studying X gene. Okay, you're studying X gene and you want to know whether this is occluded by nucleosomes or not, or or just say that you are studying a region in the genome. Okay, this is the this is the whole genome. Okay, and you have to study only this much. You have to study only this region. Okay, so for studying this region, what you do, you already have the sequence of this region, so you can design primers, the forward primer and reverse primer, only for specific for this region. Let's say this whole region is the X gene, so you can design gene specific primers and perform the qPCR. So gene specific primer, so the qPCR means quantitative PCR. qPCR means quantitative PCR. Can in this PCR we can actually quantify the amplification of the DNA. So we'll be discussing about this also in details. I'm assuming that you already know about the basics like PCR and what is quantitative PCR, semi-quantitative PCR and all. So where were we? Yeah. So you can design the gene specific primers, which will only amplify this much region. Okay. So now you want to know whether this gene, X gene, is it occluded by nucleosomes or not. So what you can do, you can, let's say this X gene is found in Drosophila. Okay, and you are studying this gene in the Drosophila. So what will you do? First, you will isolate the nuclei, isolate the cells. Okay, then isolate the nuclei, or you can directly lyse the cells, lyse the cells such as the DNA damage is minima. Okay, and you can extract the chromatin. Okay, so the first step is chromatin extraction. So you have already extracted all the chromatin. So you already have the access of the genome of the Drosophila. You already have the access of the chromatin of that certain cell type, whichever you want to study. So now you digest the chromatin by the MNase, digestion by MNase. Okay, so you have already done the perform the digestion, and this is how you get fragments. So then you perform, so after digestion by MNase you perform the gel extraction. Okay, means you can extract the DNA from this, you can extract those DNA fragments from the gel. After running the agarose gel electrophoresis, you can actually extract those DNA fragments, and that step is known as gel extraction. So once you have done gel extraction, you get the DNA fragments. Okay, so you have the DNA fragments, but those DNA, this is not normal DNA, this is nucleosomal DNA. This is already occluded by the nucleosomes. Now you can get rid of the proteins, you can get rid of the histone code. So get rid of the proteins, and then you perform the PCR, quantitative PCR. Okay, with gene specific primers. Okay, so here comes the main point, that when you perform the qPCR, the see, so for primers to go, and for primers, so this is the gene, X gene. Okay, so primers to forgo and hybridize with this template. Okay, so this is the template. So primers to go and hybridize this template, you need this gene needs to be free. If this is occluded by nucleosome, it can't go and hybridize to the template. It can't primer can't go and attach to the template, and the PCR amplification would not be efficient. Okay, so if you have nucleosomal DNA, the gene will be inaccessible, and because it is inaccessible, the amplification of gene will be low. And that's why we are performing the qPCR. If the amplification of certain gene is low, then we can definitely conclude that it is occluded by nucleosomes. So this is how we use the MNase-qPCR. But for get for make no, no, like for knowing surely that this is the case, you can also perform this MNase sequencing. What you can do after digesting DNA in the fragments, you can extract this DNA fragments, and instead of qPCR, you can perform the sequencing steps. You can prepare the library and the whole sequencing steps you can perform, and then you can know, okay, so this region was the nucleosome bound. Okay, because after sequencing you will obviously know that this region was nucleosome bounded. Why? Because all those fragments are nucleosomal DNA. Okay, so this is how the MNase-qPCR or MNase sequencing is done, and it gives us information about nucleosome positioning.